Assalamualaikum to our lecturer, Dr. Aina Nadia Abdul Halim. I am Amira Zafira Zahari with my group members Amira Binti Zulkarnain and Tiffany Junior Singgah Ana Edi from subgroup 10. We will do the presentation of our research proposal. Our research title is Biochar from Rice Husk and Utilization for Plant Crop. The content of these presentations are introduction, literature review, problem statements, objective, methodology, expected outcomes, and work plan.
Agriculture waste such as rice husk, rice straw, waste straw, sugarcane bagasse, and corn cob has accumulated approximately 2 billion tons worldwide. The residues of the agriculture waste can be converted into biochar, which has the ability to supply feedstock for biochar production and contribute in achieving long-term carbon sequestration with other useful effects on soils. Rice husk is a accomplished waste product with an average annual production of tons. However, only a tiny percentage has been used in reality, as it is believed that 90% of rice is destroyed in the open air or comes into waterways, posing substantial environmental risks. Therefore, converting the rice husk residues to rice husk biochar and implementing it as soil supplement could be a sustainable solution to rice cultivation. Biochar received attention by researchers due to its large surface area, a large number of mineral elements, a large number of functional groups, and a porous structure. Biochar is a charcoal-like substance that is created by burning organic waste in the loss or presence of oxygen in a controlled process known as pyrolysis. The morphology of rice husk biochar changes to a honeycomb-like structure with many pores forming on its surface when exposed to 500 degrees Celsius pyrolysis temperature. According to previous research, rice husk biochar has considered to boost agriculture output by enhancing soil quality and nutrient fixation. Plant growth issue. Soil salinity has been a major issue in crop production due to excessive amount of soluble salts in soil which will prevent the plant growth. Pesticides, in addition to salinity, have led to serious environmental problems which have a negative impact on soil microflora. In order to improve the physiochemical properties of biochar, researchers chemically modified the biochar. Previous research has reported on modification of biochar by using acid and alkali. Alkali treatment has enhanced the porosity and the specific surface area of biochar. A chemical reduction procedure involving reducing agents such as sodium hydroxide, potassium hydroxide, and ammonium hydroxide that is used to alkalize the biochar. Acid treatment, on the other hand, have enhanced the structure of biochar and increased its propensity to absorb polar molecules due to its hydrophilicity. The surface of biochar is oxidizing using the hydrochloric acid, sulfuric acid, nitric acid, hydrogen peroxide, and phosphoric acid in the acid modification. In this research, we have studied concentrated acid and alkali of sulfuric acid and sodium hydroxide that will be mixed with the biochar in order to modify the prepared rice husk biochar.
Due to biomass resources, it is found that rice husks are a plentiful waste item in Asia. It can be turned into biochar, but to produce a rice husk, biochar itself does not contain sufficient nutrients for crop growth, as the parts of biochar are obstructed by unpyrolized biomass and mineral ash. The objectives of this research are to prepare biochar from rice husk, to modify biochar chemically and characterize the biochar from rice husk, also to examine the plant growth. Next, I will explain the methodology. First, for preparing the rice husk biochar, the rice husk will be obtained to manufacture the biochar under a pyrolysis process. Next, it will be baked at 500 degrees Celsius to produce the rice husk biochar, and the residue and biochar will be heated at 550 degrees Celsius to make the ash. Then, they will be dissolved in 2 normal hydrochloric acids. Next, to produce a modified rice husk biochar, the biochar will be put into water and sonicated for minutes. The mixture will then be agitated and iron 3 chloride hexahydrate and ferrous chloride tetrahydrate will be added into the mixture. Next, the biochar will be mixed and stirred with distilled water, concentrated acid, specifically sulfuric acid or used alkali, which is sodium hydroxide for 24 hours. The plant's growth will be determined using the rice husk biochar and the modified rice husk biochar by putting it into the soil and thoroughly mix.
It was expected that the biochar will be successfully prepared and chemically modified from the rice husk. The chemically modified biochar with increasing levels of nutrients uptake is expected to enhance the plant growth due to the presence of the activating agents in acid and alkali modification. So this is our work plan. The first research activities is proposal writing. For proposal writing, we write from March 2022 until April 2022. For proposal presentation, we do it in May 2022. For sample preparation of biochar, we do it in April 2022 until June 2022. For the preparation of chemically modified biochar, we do it in May 2022 until June 2022. For the report writing, we do it in May 2022 until July 2022. Both submission and presentation of final report, we do it in July 2022. That's all for our presentation. Thank you for watching.
Here are our references for research proposal for this subject. Thank you for watching.